Hello everyone, my Stan. Today we will talk about the DNA replication. So this is process by which cells make an identical copy of the DNA molecule. So this is a step that made the DNA like duplicated, and there's some steps need to be done. The first step is unwinding. So there's a protein called helicase goes down the DNA strand and split into half. This is the DNA, right? These are these. And there's a protein to split them like zip, 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 like that. That's the first step. To zip it up to 2 parts. So let's go to the second step. The second step is primer. You might ask, what's primer? So primer is like each half of the DNA strand acts as a stamp, a template for a new strand. An organizer is called primers. Make a small strand of RNA called primer that acts as a starting line for replication. So that is right. This is when you zip it up like this, right? And then there's a protein come and do this at the first starting line. So this right here is the starting line, and it's where it started to continue making a new one. So the first one is synthesis. It's a third step. So it's enzymes called DNA polymerase find the primer. It will then fill the nucleus and the DNA strand to grow the DNA strand like this. That's a lot, right? And then this is a starting line. And then we add in this over here. So now we come to the fourth step. The fourth step is called RNA removal. So once strand are filled in with analyzing called executor are used as remove segment of RNA. Then a DNA polymerase fill in the holes with DNA nucleus. So let me make you understand. This is the other side, right? Because the protein only can go one side, it can't go 2 sides. So this is what I do. First is make some holes. Like this, and then go in. This. You keep do that and make some holes. And then later it will fill the holes. So it's just a little bit more complicated and a little bit different and slow as well. So of course the final step is identify segment made by DNA and the polymerase are joined together, and then by an escape called DNA reagents. Like first start with this, end up at the end this. And do you know that you need to do some things before you reach that? Because there's 4 kinds of what's it's called nucleotide. There's 4 kinds of nucleotide, and they can't all connect together. There are only 2 connected. So there's these holes. When the fourth step is when DNA is built on the small bump on the lagging strain, they are called orthogonal fragments. So these all they fill in is called orthogonal fragments. So now let's back with our nucleotide. We're going to talk about nucleotide and RNA. So first there's 4 kinds of nucleotide: A, T, C, G. So the first one is called adenine, second one is called thymine, third one is called cytosine, fourth one is guanine. The thymine have a next name, is called U. And what's it called? Let me check. I'm trying to see where I write it. Wait. Oh, uracil. That's like a name. And for an RNA, it's different than DNA, only this part. It's a connect, but in RNA, A is connected with U instead of A is connected with T, but T and U is the same. And Z is connected with G. Yeah, it's normal. Let's talk a bit more of the RNA, not that much. Let's only talk, let's not write. I'm lazy. I'm a lazy man, it's come for lazy pressure. Okay, so there's kinds of RNA nucleotide. First, there's sort of two types, DNA and RNA nucleotide. So first one is thymine. Second one, the other one have four, like the four left is adenine, uracil, cytosine, and guanine. Have four. So RNA is a single-strand nucleic acid that has various jobs include making protein and rare genes. So it's different from DNA. Instead of making protein, like storing protein and make protein and send them out, make gene protein and then send it out, they do a different job. So there are some kind of RNA. So there's about 3 kinds. So now what job it do? There's 3 jobs. First one is messenger RNA. It's called mRNA. It carries genetic information from the DNA in the nucleus to ribosome in the cytosis where protein are synthesized. So let me remind you, ribosomes is a tiny membrane organelle that combine amino acid to make useful protein for the cell to use. I'll remind you that. Second is transfer RNA, and it's called tRNA. So it's helped decode the RNA sequence into a specific amino acid sequence during protein synthesis. And the final one is ribosomal RNA. It's called rRNA. Along with the protein forms the structure of ribosome, where protein synthesis is occurs. But there's another process in DNA as well. It's a process which a segment of DNA is copied into and T molecule. So this is called transcription. So let's talk about transcription as well. So there's 3 steps to the DNA to create RNA. So step 1 is interlacing as enzymes called RNA polymerase bind to an areas of DNA called promotes. And step 2 is oligons. Is DNA unwind and explosion. A nucleotide RNA molecule copy the nucleotide on train of messenger and messenger RNA, mRNA. Step 3 is the termination. Is the RNA polymerase reach the termination signal in DNA and stop making RNA strands. So what did you learn today? Today we learned that how DNA have been reduced, mean doubled up, like make more of it, was nucleotide, like in RNA, and transcription. Hope you enjoy. Goodbye. See you next time.